Mitosis. Mitosis is also called as equational division. Now they are called as equational division because they have the equal number of chromosomes as that of the parent cell. Like this is the parent cell, and if it is having two chromosomes, suppose if it is having two chromosomes, then after the cell division that results in two cells, two daughter cells, each cell is also going to have two chromosomes only. So that's why they are called as equational division. Now usually this mitosis takes place for diploid cells. But in some cases, like in male honeybees, who has 16 chromosomes, the male honeybees develop from egg, which is haploid. Other than these exceptions, almost mitosis takes place in diploid cells. So if these diploid cells, suppose this parent cell has got 10 chromosomes, and so the daughter cells also will have 10 chromosomes each. So it's called as equational division. Phases. So during the karyokinesis, that is the nuclear division, there are the following phases. One is called prophase. So after the interphase stage of the cell, when the interphase stage is present inside the nucleus, the genetic material is in the form of thread or chromatin thread. So by the late prophase stage, these chromatin threads will condense or they become thickened to form chromosomes, in which there will be two sister chromatids attached to the center called as centromere. And the centrioles will start moving towards both the poles of the nucleus. And you cannot see any kinds of cell organelles. These centrioles will start producing the microtubules which later attaches to the chromosomes. The next phase is metaphase. During the metaphase stage, the chromosomes will be arranged at the equatorial plane. So by the end of the prophase, even the nuclear membrane disappears. So the chromosomes will be present or arranged at the center. And the centrioles at the two poles is visible, with the microtubules emerging from the centrioles. During the anaphase stage, the sister chromatids will separate and they will be pulled by the centriole microtubules towards both the poles. So they are the sister chromatids that are pulled towards both the poles. So the microtubules, which were emerging from the centrioles, were attached to a structure near the centromere, and that is called as kinetochore. So these microtubules that emerges from the centrioles, they are actually attaching to the kinetochore, and during the anaphase stage, these microtubules will be pulling the chromatids and separating them towards both the poles. Followed by this comes the telophase. Now, during the telophase stage, the nuclear membrane reappears around the chromosomes. But the chromatids are not in a condensed form, they are in the decondensed form, thin thread like. So, this is the end of karyokinesis. Followed by this, the cell will divide. So, at the end of the karyokinesis, we can see now, if you see this nucleus, there are two chromosomes in each nucleus. Here, in one cell, we can find two nuclei, and each nucleus has got two chromosomes. And when we compare with the parent cell, even the parent cell has only two chromosomes. Parent cell has got two chromosomes. Parent cell has got two chromosomes, and after the nuclear division also there are two chromosomes in each nucleus, which that is why mitosis is also called equational division. Now, followed by the telophase stage, the cells will divide, and that is called as cytokinesis. In case of plant cell, if it's a plant cell that is dividing, a cell plate develops, which later forms cell wall. A division furrow develops. That later on separates the cell into two cells, each having a nucleus.